I'm Huihuang Yan from Mayo Clinic. My presentation, the title is Epigenetic Alterations in T-cell prolymphocytic leukemia. TPL is a rare disease with a median survival of less than 1 year. So TPL itself is considered has a very poor response to conventional chemotherapy and also relapse after immunotherapy because of the required resistance. Whole genome and whole exome sequence has identified the key alteration in genetic, so including two key translocation involved genes TCL1A and TCL1C, and also recorded somatic mutation in genes including chromatin regulators and those in the JAK state signal pathway. But the epigenetic mechanism involved in this disease is largely unknown. In this study, we performed a triple sequence for 3 histone modifications: H3K4 monomethylation, H3K4 trimethylation, and H3K27 acetylation in 6 TPA cases and selenomas, and we also did the RNA sequence data for the same samples. So when we look at the profile of the cluster used H3K27 acetylation, we use the top 20,000 peak. If you look at the profile, clearly there's separation of the normal versus the TPA cases, and appears to be more loss of H3K27 acetylation in the TPA case compared to the normal. We also identified about 800 upregulated genes and about 900 downregulated genes in TPL compared to the normal, and those genes, in particular the downregulated genes, are highly enriched in the pathway related to the immune response. We further look at the correlation of the differential peak of H3K4 trimethylation and H3K27 acetylation. If you look at the table, they're more upregulated of both markers in the normal compared to the TPA cases, that indicating more loss of those 2 markers in the TPA cases. And the upregulated gene in TPL is associated with the upregulation of those 2 markers. So there's a positive correlation between the histone modification and gene expression of the nearest genes. So if we look specifically under the key gene involved in TPL, so we look at 2 ORC genes. One is TCL1A here, another one is the MEK. If we look at the RGB snapshot, in both cases, there's a gain of those histone modifications in the regulatory regions, and that are associated with the increased expression of this gene in the TPA cases, but in normal, you hardly see any signal of those 2 markers. It is a similar profile in the MEK case. So there's a gain of those 2 markers in the regulatory regions in the promoter, and also if you look at the profile here, the interaction, the enhanced region, at least in 3 of the cases, there is a gain of H3K27 acetylation in the enhanced that interact with the promoter. So accordingly, the gene of this make the more upregulated in the TPA cases. So in the opposite, if you look at the ATM, ATM is a very critical gene, and its tumor suppress has a very important role in TPA cases. If you look at here, it's the opposite trends. In TPL, the kind of loss of this mark in the regulatory region, and accordingly, there's less expression of this gene, at least in 4 of those cases in TPL. So our analysis highlights the very important role of regulatory elements in TPL and that causes the dysregulation of the nearby genes in expression.